Welcome, everyone. Today, we'll explore the fascinating journey of microbial diagnostics, from traditional culture techniques to the latest cutting-edge technologies. This evolution reflects how our tools have grown more sophisticated, helping us better identify and understand microbes. Let's start with a fundamental question we always face in microbiology. What microbe is this, and what is it doing? This question isn't just academic, it's critical across many fields. Clinically, it guides diagnosis and treatment decisions. Environmentally, it helps us monitor ecosystems and pollution. In food safety, it prevents contamination and spoilage, and in industry, it ensures quality control in biotechnological processes. The challenge is huge. Microbes are incredibly diverse. Many can't be cultured easily, and there's always pressure to get results faster and more accurately. Culture-based methods form the foundation of microbial diagnostics. They rely on isolating and growing microbes on specific media. Classic techniques include plating methods like streak or pore plating, microscopic examinations such as gram staining, and biochemical tests to differentiate species. For example, E. coli's pink colonies on MacConkey agar or Staphylococcus aureus's golden colonies on mannitol salt agar are classic hallmarks. These methods are cost-effective and allow direct antibiotic susceptibility testing, which is a major advantage. But they are slow, taking days sometimes, and not all microbes grow in culture, so they have limitations. Immunological assays use the highly specific binding between antibodies and microbial antigens. Techniques like ELISA, lateral flow assays, which many know from rapid COVID-19 tests, agglutination, and immunofluorescence allow us to detect microbes quickly. They're generally much faster than culture and can detect unculturable organisms. However, they depend on having specific antibodies, sensitivity can vary, and cross-reactivity can sometimes cause false positives. PCR revolutionized microbial diagnostics by enabling detection of unique DNA or RNA sequences of microbes. It rapidly amplifies small amounts of target DNA through cycles of denaturation, primer annealing, and extension. There are different types: conventional PCR, which you detect at the end using gels; real-time PCR or qPCR, which quantifies DNA as it amplifies; and multiplex PCR, which detects multiple targets at once. This technique is highly sensitive and specific, fast, and doesn't require live organisms. But it risks contamination, and it can detect DNA from dead microbes, which may not indicate active infection. Sanger sequencing was the first gold standard for identifying microbes at the gene level. Next-generation sequencing, NGS, took us further, allowing massive parallel sequencing of millions of DNA fragments. NGS applications include metagenomics, where we ask, who is there? By sequencing marker genes or whole genomes; metatranscriptomics, which reveals what microbes are doing by analyzing RNA; and whole genome sequencing, providing detailed typing and resistance profiles. These techniques give us unprecedented resolution and can discover unknown microbes, but they can be costly, require complex bioinformatics, and have longer turnaround times. Looking ahead, technologies like MALDI-TOF mass spectrometry identify microbes by their unique protein fingerprints, giving results in minutes. Microfluidics miniaturizes lab processes onto small chips, ideal for rapid, point-of-care testing. CRISPR-based diagnostics harness RNA-guided Cas enzymes for highly sensitive and rapid nucleic acid detection, with great potential for multiplexing. Advanced phenotypic approaches use digital image analysis to automate and speed up culture plate reading and resistance detection. Together, these represent the future of faster, more integrated, and more accessible diagnostics. There are several challenges. High costs and limited accessibility create global disparities. Analyzing large sequencing datasets requires specialized bioinformatics skills. Clinically, we debate whether detecting DNA from dead microbes reflects real infections. We also strive to balance turnaround time with thorough, actionable results and standardized diagnostics across labs. But the opportunities are exciting. Portable point-of-care devices will bring testing to patients directly. AI and machine learning will improve data analysis and resistance prediction. Integrated omics approaches will give holistic microbial insights. Ultimately, better diagnostics will support antimicrobial stewardship and strengthen global health security, crucial in pandemic preparedness. To wrap up, microbial diagnostics has dramatically evolved from simple culture plates to sophisticated molecular and digital technologies. Older methods remain important and often complement new ones, providing different layers of valuable information. The consistent goal is to deliver rapid, accurate, and actionable data that improves patient care, public health, and scientific understanding. The future is bright, driven by constant innovation and the urgent need to meet new microbial challenges. Thank you all for your attention. I'm happy to take any questions or discuss any topics further. Feel free to reach out for more resources. Also, like, share, and subscribe for more.